Okay, so guys, in the previous recording, you saw that I was explaining you about the mitochondrial evidence in order to find out the evolutionary relationship between different species. And I was actually reading this paragraph, and then the final conclusion that was made was that the greater the number of differences in the nucleotide sequences in the mitochondrial DNA, the long ago those organisms shared a common ancestor or they are distantly related. And the smaller the number of differences in the nucleotide sequences in mitochondrial DNA, the longer the more close those organisms were as far as their ancestral relationship of evolution is concerned, right? Okay, so now in order to actually understand why we are doing this topic, we will do a question, one question from your textbook and the other question from the past paper. So this is the example that is given to you in the textbook, and in which they have given you some background information about analysis of mitochondrial DNA of the different species of anole lizards that are found throughout the Caribbean and the adjacent mainland provides evidence of their relationships. Each island species of lizard is found only on one island or a small group of islands. Then on the next page, we have our table, but I'll show you the table afterwards. We have a table which shows the results of comparing the part of the mitochondrial DNA of 4 of these species. Achha, what are their 4 names? It's actually anole lizards, and the 4 species are Anolis brunnius, Anolis samaragdinus, Anolis carolinensis, and Anolis porcatus, right? Achha. The results show that the 3 species, which are brunnius, samaragdinus, and carolinensis, are more closely related to porcatus than they are to each other. And in the table which is coming up, we will be able to figure out how this can be seen by looking at the data. This suggests that these species have each originated from separate events in which a few individuals of porcatus, which means a lizard whose species name is porcatus, is spread from Cuba to 3 different places. If you remember, in the previous recording, I have explained to you what speciation is, right? Achha. You can listen to that recording also, and I'll be sharing some screenshots from the new textbook with you on the topic of evolution and speciation, right? So basically, when we say evolution has occurred, what we are trying to prove is, or what we're trying to say is actually that a new species has evolved, right? And a new species can evolve due to many, many reasons. Lots of reasons have been given to you, but the bottom line is that all those reasons should support the fact that the gene pool has changed and the allele frequencies have also changed. And a new species is arising from a previously existing species. Achha. Under the topic of speciation, 2 types have been described. One is allopatric speciation and one is sympatric speciation. The word patria is from the word patriotic, which means land, right? And allo means other. Achha. And sim means same and patria means land. So this means that if we say allopatric speciation has occurred, this means that there was a group, like there were some organisms which were living on like a geographical area, right? Then due to some catastrophe, some of those animals from the original group were separated due to an earthquake or whatever. And since they were separated and since they were now exposed to different geographical conditions, so if millions of years pass by, all these are just hypothetical assumptions, then it might be possible that those group of organisms that were isolated, they might develop some different characteristics from the original population. So this kind of a speciation in which some of the organisms, they are geographically separated from the original group of organisms, is known as allopatric speciation. Allo, other, patria, land, right? So in this example, why am I actually repeating allopatric speciation and all? Because in this example, it shows that initially there was a lizard whose species name is Anolis porcatus. it spread from Cuba to 3 different places. And since it spread to 3 different places, so the lizards from those 3 different places are a bit different from the lizard which was originally situated in Cuba. But at the same time, the differences that those other 3 species have with Porcatus are less as compared to the differences that they have amongst with each other, right? So that is why after reading this example and also after looking at the table, which is just coming up, we can deduce, or we can actually say, conclude that this kind of speciation is allopathic. In exams, you will be given examples like these, and sometimes you might be given data or you might not be given data, and you have to decide for yourself which kind of speciation has occurred. If you made a wrong decision, then of course you will lose marks, because most probably what happens is they give you a piece of information, they ask you to decide which kind of speciation has happened, and in the part B or C of the question, they ask you to describe and explain how that particular kind of speciation happened. So this means that if you have chosen the wrong answer, like sympatric instead of allopatric or vice versa, then you will be losing marks, right? Let's look at this table. This table, and this, you should know how to read this table, and I'll tell you how we can do it. Now just look here. We are supposed to compare the part of the mitochondrial DNA of 4 of the species of these anole lizards, and these numbers are actually giving us the differences between the base sequences of the mitochondrial DNA between 2 species. For example, there is a number which is 12.1, right? Now, this number shows you that this is the number of the differences between the mitochondrial DNA of Brunnius and Samaragdinus, right? 16.7 is the number of the differences between the species Brunnius and Carolensis. 11.3 is the number of the differences in the base sequences of mitochondrial DNA of species Brunnius and Porcatus. If we keep on comparing these differences, we will see that every species, which means Brunnius, Samaragdinus, and Carolensis, their differences with each other are much, much more as compared to their differences with Porcatus. See, the difference in the base sequences of Brunnius and Porcatus is 11.3, which is a small number, right? The difference between the base sequences of Samaragdinus and Porcatus is 8.9, and Carolinensis and Porcatus is 13.2. So all this shows that these 3 species of lizards are not so closely related to each other as much as they are closely related to all of these 2 Porcatus, showing that initially this species Porcatus was living on an island in Cuba, as can be seen over here. And then maybe allopathic speciation occurred due to any geographical reason, due to which these lizards were isolated from the original population. They settled in different places and hence they acquired some differences. But then at the end of the day, they were similar to their original species, right? So this is what they are trying to explain. Let us do some other example. This example is from a past paper in which it's about a fruit fly, Drosophila. It has many different species. 3 of these species, Drosophila pseudo-obscura, Drosophila persimilis, and Drosophila miranda, are thought to be closely related. Samples of these 3 species were collected from the western United States of America. Figure 5.1 shows where these species naturally occur. So you can see where they are naturally occurring. These 2 species are occurring on this thin belt, and this species is occupying this entire part of the United States, right? Let's see. The base sequences of the 4 regions of the DNA of each species were sequenced. The divergence of these base sequences, which means the differences, right? The divergence means the differences of these base sequences in Drosophila pseudo-obscura and Drosophila persimilis from the sequences in Drosophila miranda were calculated, right? Let's go back. Miranda and persimilis are over here, and this is in a larger quantity, which is occupying this part of the map, right? Okay, so with reference to the table 5.1, describe the events that Drosophila miranda, sorry, not the events, with reference to table 5.1, describe the evidence with reference to this table, describe the evidence that miranda may be more closely related to persimilis than to pseudo obscura, right? Again, I'm repeating, with reference to table 5.1, describe the evidence that Drosophila miranda is more closely related to persimilis than to Drosophila pseudo obscura. Now let's compare the differences. Drosophila miranda, these are all the percentage divergence of the base sequences from that of miranda, right? Let's compare. And 4 different DNA regions have been used. The differences between miranda and persimilis are less. Look here. I'll just shade these. This one, this one, this one, and this one, right? The differences or the percentage divergence of the base sequence of miranda from each of these species has been listed down over here. So the difference between, or the differences of the base sequence between miranda and persimilis is 2.4, whereas between miranda and pseudo obscura is 2.5. The differences between miranda and persimilis is 7.3, and the differences between miranda and pseudo obscura is 8.1, and so on. So this means that if we look very closely at the table, we can conclude that the Drosophila miranda is more closely related to Drosophila persimilis because the percentage divergence of the base sequence of each of the 4 regions of DNA is less between miranda and persimilis as compared to between miranda and pseudo obscura. And this is the answer from the past paper. It says divergence values are less for persimilis than for pseudo obscura at all 4 DNA regions. And this says, or reverse argument. You can give a reverse argument about pseudo obscura, that their values are much more. It's the same thing. And then you can also use figures to support your answer, right? Now there is another question. And this question is, as I told you, whenever you will be given such kind of questions, they are always related to the concept of speciation, in which you have to decide whether it is allopatric speciation happening or sympatric speciation happening. And then after you've decided, then you have to also list down how it happened, right? The area where Drosophila pseudo obscura is found, is separated from the areas where the other 2 species are found by a high range of mountains. This in itself is giving you a hint that allopatric speciation has occurred, because in allopatric speciation, the 2 group of organisms are separated by a geographical boundary. So explain how the species Drosophila pseudo obscura could have evolved from a population of Drosophila miranda. So let me tell you that you always have a generic answer for such kind of questions. Number 1, allopatric speciation has happened due to the fact that there was a geographical or physical barrier, which led to a stoppage of the gene flow between these 2 populations, and hence maybe random mutations were also happening at the same time. So since these 2 populations were separated geographically and there was no interbreeding or gene flow between them, so, and also at the same time they were exposed to different environmental conditions, so we expect different selection pressures to operate on them. And over a period of time, millions of years, again I would say these are all hypothetical assumptions, changes in allele frequencies, they are taking place, so that the gene pool changes and the advantageous alleles are passed on, genetic drift is taking place, and the Drosophila obscura, what's the name? Drosophila pseudo obscura becomes genetically isolated, which means it becomes genetically different from the original population, which was miranda. Let me tell you one thing, guys. In such kind of questions, as I said, the answers are always generic. So what are those generic terminologies that you will always use while answering such questions is whenever you talk about speciation, number one, you will decide, the first step should be that you should decide which kind of speciation is happening. After you've decided, of course, you will see that there is no gene flow. Mutations are happening at the same time randomly. Organisms are geographically separated, so they are exposed to different environmental conditions, thereby changing the allele frequencies, causing genetic drift. Since they cannot, these two group of fruit flies cannot interbreed, so it leads to finally the reproductive isolation of one of them. In this case being Drosophila pseudo obscura. So this is how you will solve such questions, and I would advise all of you to search for the similar kind of questions which are based on you identifying what kind of speciation is taking place, and also at the same time you should be fully capable of reading such kind of tables so that you can know how to read data, analyze data, and make valid deductions and conclusions, right? So I'm stopping my recording over here, and I'll be sharing some more recordings with you. Allah Hafiz.